Hello everyone, welcome back to Metabolism in Biochemistry. In this lesson we're going to talk about gluconeogenesis. Now this is following the last two lessons, which was about glycolysis. It has some similar concepts in terms of molecules because this pathway is quite similar to glycolysis, except there's a few changes that we're going to talk about. And after this video, we're going to discuss about regulation in glycolysis and gluconeogenesis. So what is gluconeogenesis? Unlike glycolysis, where you broke down glucose into pyruvate molecules, in this case, we're going to form glucose from scratch. And that means we're going to use non-carbohydrate molecules to form glucose. And that includes many types that we're going to talk about. But before we get into that, let's talk about the purpose of gluconeogenesis. Why do we want to use this pathway? Well, firstly, there are some tissues in our body that need glucose. They rely on glucose. And this includes the brain and some other tissues. And in order to have glucose available at all times, the liver has to maintain the blood glucose levels. So the first thing is to maintain the levels of blood glucose. So maintain glucose levels. And this is helpful for tissues such as the brain, where its only way is to use glucose. And then we have like red blood cells, where they do not have mitochondria. They have to use anaerobic respiration, so they have to use glucose. Secondly, in other tissues that are not the liver and not kidneys, because kidneys do also maintain blood glucose levels, but to a lesser extent. In other tissues, we need to go back towards glucose, where we eventually get glucose 6-phosphate. We need to do that so we can use it in other pathways, such as in the pentose phosphate pathway. And in those tissues, the purpose is that you can use glucose 6-phosphate for metabolite purposes. So you can produce metabolites that can be used in other pathways. So in other tissues, we can have metabolites. And another purpose is to balance energy. When you have a lot of energy, you want to go back to glucose 6-phosphate, where it can be stored as glycogen, such as in muscle. So we have energy balance. And there's probably other reasons too, but these are the three main purposes. And in different tissues, there are different purposes for this pathway. So now let's get into some non-carbohydrate molecules. So firstly, we have lactate. Now lactate is used in active muscle in order to produce energy. It is used anaerobically because in active muscle, where you have a lot of action going on, your aerobic pathway is overly exhausted with all the work it has to do. So your body switches to anaerobic respiration, and that leads to the production of lactate. And this lactate can go back to the liver, where it can be turned back into pyruvate. And then from then on, it can go to gluconeogenesis and you get glucose from it. And that is the Cori cycle, where you get lactate, it goes back to the liver, it turns into glucose, goes back to the muscle, and it's a whole cycle. Another thing is alanine, which is a byproduct of muscle breakdown, and there could be some other amino acids as well, but alanine is one of the main ones, and you can also get it from the diet. And lastly, we have glycerol. Glycerol is a product from triglyceride breakdown. So when you break down fats, you get glycerol and fatty acids. Fatty acids can be oxidized to form acetyl-CoA, which can be used in the Krebs cycle. However, glycerol can turn into dihydroxyacetone phosphate and enter gluconeogenesis to produce glucose. Moving on, thermodynamics for gluconeogenesis is interesting. In glycolysis, the delta G is negative 90 kilojoules. And you might think that gluconeogenesis, which is going backwards, is going to be a reversal, right? But well, no, it isn't. Because if it were the reversal, it would have a delta G of positive 90 kilojoules, which is highly unfavorable and non-spontaneous, while gluconeogenesis actually has a delta G of negative 48 kilojoules. And why is that? That is because we do not use the same enzymes for the irreversible steps. Now, if you recall, steps 1, 3, and 10 are irreversible. And when we say irreversible, of course, there's some reactions that go backwards, but it's highly non-spontaneous for going backwards in glycolysis with the enzymes at steps 1, 3, and 10. However, in gluconeogenesis, we have other enzymes that catalyze those steps to go backwards. So for example, when you go from glucose to glucose 6-phosphate, you use a glucokinase in the liver, it's isoform. However, going backwards from that, it's non-spontaneous. However, in gluconeogenesis, we have an enzyme, and this is kind of like a bypass enzyme. And it's an enzyme called glucose 6-phosphatase. And it is actually spontaneous. So if both glycolysis and gluconeogenesis are spontaneous, how does that make sense, right? Well, what the difference is how much of the enzymes are expressed so that you follow either or pathway. Both pathways are always active at the same time. However, whichever pathway is more active is what matters. And that's where the cell goes towards. So now moving on, now that we understand the basics, let's move on to the actual reactions. So here we have all these structures and all the reactions taking place. We have 4 separate steps. So for reactions, in some textbooks, reactions 1 and 2 here. So 1 and 2 are kind of in the same step, except it has 2 reactions, but I made it into 4, so it's more simple and easier to digest. And we're going to begin with pyruvate. So firstly, we have to know that lactate and alanine, they convert into pyruvate. And that happens in the mitochondria. So this is the mitochondria. And lactate and alanine. So let's have lactate and alanine. They convert into pyruvate in the mitochondria. Now, this pyruvate, the first thing it does is it converts into oxaloacetate. And this is done by pyruvate carboxylase. So, it is catalyzed by an enzyme called pyruvate carboxylase. And from the name, a carboxylase adds a carbon dioxide group. Adds a carbon, and here we have pyruvate, and we're basically adding, so this is the equivalent, so this is this. And to that we're adding a carbon dioxide. So in this reaction, we're adding in carbon dioxide, so CO2. We're also using ADP as an energy source. So we're using ADP, and we're going to get out of it ADP plus PI, and also we're going to use water. So I'm just going to write it here because there's no space. So we're using water, and we also get out of it plus 2 hydrogen ions. We're not going to get into that, but this is basically from that we get oxaloacetate. And now oxaloacetate should technically go on to become phosphoenolpyruvate, but the thing is, phosphoenolpyruvate should be in the cytoplasm because the reaction takes place in the cytoplasm. However, oxaloacetate cannot cross the mitochondrial membrane. However, malate can. So what happens is oxaloacetate converts into malate so that malate can go out, and then this malate actually converts back to oxaloacetate. So you basically have this molecule that just works to transport oxaloacetate. And the enzyme that catalyzes this reaction is called malate. So malate dehydrogenase. It's called malate dehydrogenase. And what this does is, it is a reversible enzyme, so you can go both ways. This enzyme can reverse the reaction. That is the reaction we see here, where it's reversed, so it's the cytosolic malate dehydrogenase. And you can see here, so this is oxaloacetate, and here is a ketone, right? So here, this would be a ketone, and this is the oxygen of the ketone. And here, instead of a ketone, what do we have? We have an alcohol group. What happened is that oxaloacetate was reduced to, the ketone was reduced to the hydroxyl group. The ketone was reduced to a hydroxyl group. And this is malate. This is the structure of malate. And malate goes out through its own transporter, and then malate is converted back into oxaloacetate by the same enzyme except it's cytosolic. And then oxaloacetate finally, which is the second reaction, and in some textbooks both of them are the first reaction and it's just step 1, step 2. However, I put it this way. And with the help of an enzyme called phosphoenolpyruvate, so I'm going to write as PEP, phosphoenolpyruvate, and this is a carboxy kinase. So with the help of carboxy kinase, what we did is, not only did we remove the CO2, right, we also added a phosphate, because now it's phosphoenolpyruvate. That phosphate came from hydrolyzing GTP into GDP. And the phosphate is incorporated into this, and we have the structure of phosphoenolpyruvate. So what we did is we removed the carbon dioxide, and the oxygen became phosphorylated as well. There was a phosphorylation reaction because there was a kinase, phosphoenol, because the oxygen is bonded to a phosphate, and the bond went to form between the carbon and carbon here. So, and that is between these carbons. And then we got the structure of phosphoenolpyruvate. And then from then on, we have a series of reactions. So I didn't really show them, but it's between phosphoenolpyruvate to fructose 1,6-bisphosphate. Remember, in glycolysis, the steps from fructose 1,6-bisphosphate to phosphoenolpyruvate were all reversible reactions, right? If the cell is favoring gluconeogenesis, what's going to happen is glycolysis enzymes are going to be regulated to be inhibited, and the concentrations of downstream elements are going to increase, and an equilibrium is going to favor it towards going in this direction. So those reversible steps depend on the concentration. So when concentration of phosphoenolpyruvate increases, the equilibrium favors towards the other molecule. And then eventually, after a series of steps, you get fructose 1,6-bisphosphate, which is the third reaction in glycolysis. And this was irreversible. So in this case, we have another enzyme to bypass this. So here is the structure of fructose 1,6-bisphosphate. So this is a fructose. We have a phosphate on the first and the sixth carbons. And what happened was DHAP and GA3P, it got converted into fructose 1,6-bisphosphate, right? What glycerol does is it enters into gluconeogenesis at this step, well, the one before this, which is it gets converted through a series of reactions into DHAP, and DHAP combining with J3P, together they make fructose 1,6-bisphosphate. And now, to go back to fructose 6-phosphate, remember this step, the third step was the major control point of glycolysis, because after this, it was committed to give us pyruvate. In this case, we don't have phosphofructokinase, and instead, we have an enzyme called fructose 1,6-bisphosphatase. It's a bisphosphatase. So what it does is it cleaves the phosphate group. So now you're removing the phosphate from the first carbon. And here we're actually adding water into it. So we're adding H2O. And we're releasing an organic phosphate. Now, remember the second step. This is the second step in glycolysis, if you look at it backwards. And in this step, remember it was a reversible reaction. As now I'm showing an arrow here, but it's a reversible reaction. And in glycolysis, it was the other way, right? So now we're looking at it the other way. And when fructose 6-phosphate concentrations increase, obviously equilibrium is going to favor towards the other side, right? So if this increases, it's going to go to the reaction will go towards glucose 6-phosphate. And now it goes towards glucose 6-phosphate, which is molecule right here, and we have the phosphate at the sixth carbon. From that, we get glucose. we do the fourth reaction. And the fourth reaction does not happen in every tissue. It only happens in the liver and the kidney to a lesser extent, because those tissues are in charge of keeping the balance of glucose in the blood. Because if glucose 6-phosphate is dephosphorylated to become glucose, which is the free form, so free, it can go into the bloodstream. And if every tissue did this, you would just have this uncontrollable amount of glucose in the blood. So the tissues like the liver and kidney are responsible for this. So they are the only ones that have this enzyme to convert glucose 6-phosphate into glucose. So you might wonder, now what is the purpose of gluconeogenesis if they're not going to get glucose, right? However, in some tissues, they have other purposes as we discussed before. So in muscle, this is going to go through glycogenesis. So you'd have this go to glycogenesis. So in different tissues, you have different needs. And according to cellular needs, where the regulation, allosteric, covalent, those dictate which pathway is favored. And we're going to talk about regulation in the next one or few videos. However, in liver and kidney, let's talk about what happens to get the free form of glucose. The enzyme, first of all, you should know that catalyzes this is called glucose 6-phosphatase. So it's a phosphatase. It removes a phosphate group. However, this does not just happen in the cytoplasm. And I have this diagram here to talk about it. So assuming here is the cytoplasm. Of course, the borders are not there, but just assume that's the cytoplasmic side and this is the ER side. So actually, we have this transporter which is called T1. So I'm going to write it right here, T1. And glucose goes through this into the ER. So you have glucose in the ER. So glucose. And this is glucose 6-phosphatase. So let's put an arrow. So we know this. And it is a protein. It's not a transmembrane protein, it's an integral protein. And it stays there to convert glucose 6-phosphate. And apologies, I say glucose here, it's glucose 6-phosphate. So let's change it to glucose 6-phosphate. And this converts into glucose. It converts into free glucose. And you release a phosphate, so you have the release of phosphate. And glucose and phosphate, inorganic phosphate, they go back into the cytoplasm with these two transporters. So we have the T2 transporter, which T2, this is T2, the green one is T2, and T2 helps phosphate go back into the cytoplasm. And then we have T3, which helps glucose go back into the cytoplasm. And now glucose is back into the cytoplasm and it can use GLUT transporters to go back into the bloodstream to maintain blood glucose levels. And that is covering all these steps of the gluconeogenesis pathway. So now let's just talk about the overall results of it by going down here and discussing it. I forgot to mention here that when we used malate dehydrogenase, we actually used NADH. So here we converted NADH into NAD, and that is where we got one of the hydrogens from. So we added the hydrogen here to the ketone, and we also have to add a hydrogen here, which I'm not showing the hydrogens, but so that's just from H+ ion. However, we use an NADH, and then to convert malate back into oxaloacetate, we have to oxidize malate. So here, which NADH is a reducing agent, it reduced malate by giving the hydroxide. And here we do the opposite. We do the opposite because we're going from malate to oxaloacetate, and malate is more reduced. So in this case, we remove a hydride. And well, how do we do that? Well, we actually just use NAD, and from that we get NADH. So we basically don't lose any energy because we're getting it back. And then NADH can be used in the reversible reaction of the 6th step in glycolysis to form a molecule that is necessary. Because as you recall, in glycolysis, we used NAD to make NADH, and that was going down glycolysis. However, as we said, the reversible reactions, we actually go down through the glycolysis pathway, and we can go back by using NADH and converting it into NAD further down the gluconeogenesis pathway. Now let's look at the overall reactants and products. So keeping in mind that we do everything twice until the fructose 1,6-bis phosphate, because you need 2 pyruvates to make 1 glucose. Remember, we used 1 glucose to break down into 2 pyruvates in glycolysis, so we do the opposite now. We need 2 pyruvates because each pyruvate has 3 carbons, and glucose is a 6-carbon molecule, so we need 2 pyruvate molecules. So everything we do it twice. So beginning with pyruvate, so we have 2 pyruvates plus we have 4 ATP molecules. The ATPs come from the glycolysis pathway where we use the reverse reactions. Remember, we made 4 ATPs in the payoff phase, while this, we're spending 4 ATPs to go backwards through some of the reactions that require ATP. And then we also used 2 GTPs, which was actually, it was used in this step, in the 2nd step, which converting oxaloacetate to phosphoenol pyruvate. And then we used 2 NADHs, which is also used in one of the reverse reactions. And we also use 6 water molecules, which you don't really need to know them. And to produce, we produce glucose, which was the final goal in gluconeogenesis, as the name says. And then you can have 4 ADP, right? Obviously, when you use ATP, you produce 4 ADPs. We produce 2 GDPs. And obviously, the phosphates that we lost is 6, which is 6BI. And also 2 NAD pluses and 2 H plus ions. And we had said before, the whole of gluconeogenesis is spontaneous, and depending on if you have the enzymes for it, it will go off. It will happen spontaneously because it is favorable, because delta G is negative 48 kilojoules. So delta G is equal to negative 48 kilojoules.